Now that we've finished learning about the structure and chemistry of individual amino acids, it's time to look at what happens when amino acids join together and form a polypeptide like the one that we see here, and how this polypeptide undergoes 4 different transformations in order to form ultimately into a fully functional protein. So the first of these transformations is actually the formation of this particular structure here. This is what we call the primary structure of a polypeptide, and we can see it basically exists in a straight line that is read starting with the N terminus typically on the left and ending with the C terminus typically on the right hand side. And all that the primary structure is designed to show us is the order or the sequence in which the amino acids join together. And the order is very, very significant because the order depends on which R group is placed in which specific location. So remember that the amino acid R groups are anything that is independent of the main chain of the polypeptide. So everything that I've circled here are all examples of different R groups of the amino acids that we have. Now, just as a review of reading what a polypeptide structure is like, again, typically we start with the N terminus, which is why it's located on the right-hand side, because in English we read left to right. Now, if we start at the N terminus, we can make our way through it and determine the number of amino acids that are found in this primary structure simply by counting the repeating nitrogen, carbon, carbon, NCC, 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 etc. And for every NCC repeat that we have, that is a single amino acid. Now, as you read NCC all the way from the N terminus, and when you make it all the way to the C terminus on the right-hand side, what you are reading is the backbone of the polypeptide. So the backbone here, you can look at it as the repeating NCC, NCC, NCC units, or you can look at it as every part of the polypeptide that doesn't include the R groups. Now, even though the R groups aren't part of the backbone, the order of amino acids is effectively looking at the order of which R group comes first, second, third, etc., because this is going to affect the final structure of the polypeptide, because depending on whether you have a nonpolar or polar, acidic, or basic R group, this is going to affect the way that the protein folds in on itself and therefore affect what the final structure is going to look like. And therefore, the order in which the R groups fall is very important in determining the structure. Now, after the primary structure has been formed, the backbone and the R groups undergo a sequence of movements that results in the formation of a 3D structure, and the first of which is what we call the secondary structure. Now, the reason why the primary structure will actually fold over itself is simply because amino acids are polar, even if they don't have a polar R group. If we see here, we can look at the backbone specifically and analyze the polarity that we find. So any remnants of the amine functional group, the nitrogen is going to have a negative pole, while the hydrogen is going to have a positive pole. And conversely, the remnants of the acid functional groups, the double bonded oxygen is going to have a negative pole, while the carbon bonded to the double bonded oxygen is going to have a positive pole. And these oppositely charged poles can form hydrogen bonds with each other and cause the backbone in order to twist into 2 commonly formed shapes. The structure that we see on the top kind of forms what we see as a spiral, and another word for a spiral that we use in biology is the word helix. And since this is the most common form of secondary structure that occurs, and also the first one discovered, we call this an alpha helix. And we can see this spiral shape forms because of hydrogen bonding that exists between the polar groups that are found on the backbone. The second most common type of secondary structure that was discovered is where the backbone folds over itself in multi-layered sheets like this. So if the polar groups cause the backbone to fold over itself, as we see here, we get a structure that is called a beta sheet. And if we look at an entire polypeptide structure, we can see multiple examples of alpha helices. Remember, if you don't know how to spell this word, helices is the plural form of helix, and then we can see an example of a beta sheet forming here. Although if we look at this protein forming here, we see several examples of the structure that are folded in neither an alpha helix nor a beta sheet. So what exactly is going on here? Well, remember that alpha helices and beta sheets simply form because of hydrogen bonds that exist exclusively in the backbone, which means we haven't taken a look at the R groups yet, and that, if we do that, brings us to tertiary structure. So unlike in secondary structure, which is exclusively formed by hydrogen bonding that exists within the NCC backbone of the polypeptide, tertiary structure looks at different types of interactions that occur between the different R groups of a polypeptide. And the most obvious example of tertiary structure folding occurs when you have a combination of polar and non-polar R groups that exist. For example, if we have polar side chains, so one example of an R group that has a polar structure is any R group that has an OH group here, because this can then form hydrogen bonds with water. So remember that most of your body is made out of water, and water has the ability to form hydrogen bonds with polar groups. However, nonpolar molecules do not have the ability to form hydrogen bonds, and therefore they do not want to be near water because they are hydrophobic. So if we look at this primary structure here folding into a tertiary structure, we can clearly see that all of the nonpolar side chains or nonpolar R groups are moving away from water on the inside of the protein, because again, they can't form hydrogen bonds, which means that they don't have the ability to attract to water the way that polar R groups do, and instead they will all congregate on the inside of the protein and hold themselves together using dispersion forces, which are the only intermolecular force that nonpolar molecules can make. Now, conversely, if we know that polar R groups can form hydrogen bonds with water, we would accept that any polar R group would be found on the outside structure of a protein so that they can form hydrogen bonds with water and stabilize the protein like that. However, we can recognize that not all R groups are simply polar or non-polar. We also have more specialized R groups, such as the acidic and basic R groups. So remember that when acids ionize in water, a water molecule is going to pull the H plus off of the acid and give the oxygen here a negative charge, whereas a base, an amine functional group, is going to do the opposite. The nitrogen is going to pull a hydrogen off of water to make OH minus, but the extra hydrogen will make this nitrogen positively charged, and when you have a basic R group that is positively charged in one location, it can attract to the negative charge of an acidic R group in a different location, and because this is actually a full positive charge and a full negative charge attracting to each other, we can actually have an ionic bond that forms, and we can see in this structure, we can see an alpha helix here and an alpha helix here, and these two alpha helices are pulled close together because there's a basic R group on one and an acidic R group on the other that is going to attract to each other and hold them together. Now, the final R group, or the final R group interaction, rather, that we can talk about is from a specific amino acid called cysteine, which has an SH group. This is what we call a thiol group in organic chemistry, and sulfur-containing compounds undergo a very specific reaction when the tertiary structure is formed. Each of these sulfur-containing groups are going to have their hydrogens removed, and they're going to be replaced with a bond between the two sulfurs like this, which is a covalent bond that is what we call, let me fix the spelling here, a disulfide bond. Disulfide meaning that it is a bond in which two sulfur molecules are connected using an actual covalent bond, meaning that the electron sharing exists. And we can see that if this if this polypeptide has a cysteine R group here and a cysteine R group here, the disulfide bond is going to connect these two parts of the polypeptide together. So we can see from tertiary structure, it is easily the most complicated because it involves every possible different example of inter and even intramolecular interactions. Now, the final type of structure is what we call a quaternary structure, and although not all proteins have a quaternary structure, it's still important to talk about because many of the most important proteins in the human body have quaternary structures, and these form simply when we get multiple, meaning more than one polypeptide structures that form together. So what we see here in each of these, we can see that each of these quaternary structures are color-coded, and each different color represents a tertiary structure that is actually a physically separate molecule. So this molecule here is one of the most important proteins in the body. This is insulin, which is a hormone that regulates your ability to absorb glucose from your blood into cells so that your cells can produce energy. And we can see that insulin is actually made of 6 different tertiary structures that are all joined together and held together by a non-protein component in the center. This molecule here is hemoglobin, almost literally the most important protein in the body, because hemoglobin's job is to transport oxygen in your red blood cells. So not having hemoglobin form by connecting 4 different polypeptides together means that you would die in minutes if you couldn't transport oxygen. And this protein here is a rather interesting structural protein called collagen. This is what your tendons and ligaments that anchor your bones and muscles together are made out of. And we can see collagen, rather simply, is made of 3 different polypeptides that are actually 100% alpha helices. In the next video, we're going to look at how the different forms of polypeptides are used in the body in order to give us the different functions that proteins take within organisms.